Assalamu alaikum. I am Reha Parvez from BSBT, and I'm here to present the topic fingerprint details and detection techniques. First of all, I will explain what fingerprints are. A fingerprint is an impression left by the friction ridges of a human finger. The recovery of partial fingerprints from a crime scene is an important method of forensic science. Moisture and grease on a finger result in fingerprints on surfaces such as glass or metal. Deliberate impressions of entire fingerprints can be obtained by ink or other substances transferred from the peaks of friction ridges on the skin to a smooth surface such as paper. Fingerprint records normally contain impressions from the pad on the last joint of fingers and thumbs, though fingerprint cards also typically record portions of lower joint areas of the fingers. Human fingerprints are detailed, nearly unique, difficult to alter, and durable over the life of an individual, making them suitable as long-term markers of human identity. They may be employed by police or other authorities to identify individuals who wish to conceal their identity, or to identify people who are incapacitated or decreased and thus unable to identify themselves as in the aftermath of a natural disaster. Next, we have types of fingerprints. There are three basic types of fingerprints: whirl, loop, and arc. Next, fingerprint identification. How we can identify a fingerprint. Fingerprint identification, known as dactyloscopy or handprint identification, is the process of comparing two instances of friction ridge skin impressions from human fingers or toes, or even the palm of the hand or sole of the foot, to determine whether these impressions could have come from the same individual. The flexibility of friction-rich skin means that no two finger or palm prints are ever exactly alike in every detail. Even two impressions recorded immediately after each other from the same hand may be slightly different. Fingerprint identification, also referred to as individualization, involves an expert computer system operating under threshold scoring rules, determining whether two friction-rich impressions are likely to have originated from the same finger or palm or toe or sole. Next, an intentional recording of friction ridges is usually made with black printer's ink rolled across a contrasting white background, typically a white card. Friction ridges can also be recorded digitally, usually on a glass plate, using a technique called live scan. A latent print is the chance recording of friction ridges deposited on the surface of an object or a wall. Latent prints are invisible to the naked eye, whereas patent prints or plastic prints are viewable with the naked, unaided eye. Latent prints are often fragmentary and require the use of chemical methods, powder, or alternative light sources in order to be made clear. Sometimes an ordinary bright flashlight will make a latent print visible. Next, when friction ridges come into contact with a surface that will take a print, material that is on the friction ridges, such as perspiration, oil, grease, ink, or blood, will be transferred to the surface. Factors which affect the quality of friction ridge impressions are numerous: playability of the skin, deposition, pressure, slippage, the material from which the surface is made, the roughness of the surface, and the substance deposited are just some of the various factors which can cause a latent print to appear differently from any known recording of the same friction ridges. Indeed, the conditions surrounding every instance of friction ridge deposition are unique and never duplicated. For these reasons, fingerprint examiners are required to undergo extensive training. The scientific study of fingerprints is called dermatoglyphics. Dermatoglyphics. Next, usage of technology. Now we will discuss what is the use of technology in identifying fingerprints. Technology is quickly taking over every aspect of our lives, including solving crimes. The rapid improvements in technology have meant that solving crimes almost takes on a futuristic factor, like something from a work of fiction. During the forensic science process, forensic equipment is used to process samples and evidence to solve crimes. Measurements include analysis of evidence, fingerprinting, or DNA identification, analyzing drugs or chemicals, and dealing with body fluids. Importantly, it's the fusion of science and technology that allows forensic scientists to do a lot of their work. Especially sciences such as biology, chemistry, and mathematics are combined with various technologies to process evidence. There are many different technologies used in the forensic sciences that most people don't know exist. Some of these technologies include desktop SEM, alternative light photography, digital surveillance for gaming equipment, facial reconstruction, DNA sequencing, automated fingerprint identification, and many more. These are only few. First of all, we will discuss Phenom Desktop SEM. The Phenom SEM is the best tool for forensic scientists as it offers a very simple to use, fast and high quality imaging tool with the added capability of determining elemental composition. This assists scientists in analyzing evidence quickly and reliably to help solve criminal cases, especially the Phenom GSR. Desktop SEM allows crime labs to search for gunshot residue particles. It's automated and the software and hardware are usually integrated, so it's very user friendly. The Phenom GSR desktop SEM also uses a unique system that combines automated GSR analysis and classification software with element identification and the easy to use Phenom Excel desktop SEM. There are lots of software parameters which can be adjusted to individual needs. For example, you can optimize it for sensitivity or speed. Second one is alternative light photography. Alternative light photography is one of the quickest ways to detect whether damage has been done to a body. Sorry, before it even surfaces on the skin. Used by forensics nurses, alternative light photography can sometimes mean the difference between life and death. The camera uses blue light and orange filters to see whether bruising has occurred below the skin surface and ultraviolet light to enhance bruises. Bite marks in surface for trace evidence. Infrared photography is used to enhance blood that's difficult to see on dark and patterned clothing and tattoos due to decomposition, lividity, and burning. Next one is digital surveillance for gaming equipment. Criminals sometimes hide illicit data on an Xbox in the hope that a gaming console won't be seen as a likely evidence target. With an XFT device, once the Xbox file system is mounted, an analyst can browse the directory tree, list its contents, open and view files, and expand subdirections and files. The XFT device will give authorities access to hidden files on Xbox hard drives and can also record access sessions to be used as evidence in court. So the criminals can easily be found. Next one is facial reconstruction. Facial reconstruction is a method used in the forensic field when a crime involves unidentified remains. The process recreates the face of an individual from their skeletal remains through a combination of artistry, anthropology, osteology, and anatomy. There are three main types of facial reconstruction: two-dimensional photographic prints are drawing, three-dimensional sculpture are high RES 3D computer image, and superimposition. While not always the most reliable technology, facial reconstruction is used by many forensic labs to determine the appearance of victims who are too damaged or whose bodies are decomposing to make a visual identification. The user inputs data into the software and a possible physical appearance is deduced. Next one is DNA sequencing. DNA is used to identify both criminals and victims by using trace evidence such as hair or skin. DNA sequencing determines the order of the four chemical blocks called bases that make up the DNA molecule. Although DNA evidence alone isn't enough to secure a conviction today, DNA profiling has become the gold standard in forensic science since the first case more than 30 years ago. DNA sequencing allows forensic scientists to sequence STR, short tandem repeat markers, potentially resulting in an increased ability to differentiate between individuals in complex DNA mixtures. Additionally, alternate marker types such as SNPS, single nucleotide polymorphisms, can be more easily integrated into casework laboratories, resulting in new capabilities such as ancestry or phenotype prediction in unsolved cases. Next one and last one is automated fingerprint identification. Fingerprint identification allows forensic scientists to compare fingerprints found to an extensive digital database. Since fingerprint identification first emerged in the 1980s, automated fingerprint identification systems have become central to the work of police and other law enforcement agencies around the world. but dramatically increasing the potential for successful identification of a suspect. These systems have fundamentally changed the way that authorities approach an investigation of a wide range of criminal activities. The rapid adoption of AFIS has inevitably led to further investment in developing it. Enhancements include the introduction of palm prints, interfacing the AFIS with other criminal justice information systems, interfacing with digital mugshots and live scan devices, and the use of multi-modal biometrics. Newer technologies such as magnetic fingerprinting thus also means investigators are able to get a perfect impression without compromising the fingerprint. Next, fingerprinting dead humans. The human skin itself, which is a regenerating organ until death, and environmental factors such as lotions and cosmetics pose challenges when fingerprinting a human. Following the death of a human, the skin dries and cools. Obtaining fingerprints from a dead human to aid identification is hindered by the fact that only the coroner or medical examiner is allowed to examine the dead body. Fingerprints of dead humans may be obtained during an autopsy. Next, chemicals used in fingerprinting. Now we will discuss the chemicals which are used in fingerprinting. Forensic scientists rely on 4 primary chemicals to relieve and collect fingerprints: iodine, cyanoacrylate, silver nitrate, and ninhydrine. These chemicals react to substances within the fingerprint, such as oil and sweat, making the print change color so analysts can see it better. Forensic scientists usually use iodine and cyanoacrylate, better known under the brand name superglue, by heating them to create fumes. They use silver nitrate and ninhydrine as a spray or dip. Other chemicals used for fingerprinting are diazofluorine 1 or DFO1, rhodamine, drops, sodium block, phenol, europium gelato or TEC, and acid fusion. Next, these chemicals help develop fingerprints on more porous surfaces or enhance prints already produced by other chemicals by dyeing and stabilizing the prints. Other ways are blood evidence. Fluorescence causes a chemical reaction between oxygen and the hemoglobin present in blood. This chemical is useful for fine blood stains and smears, which may appear in different crime scenes. Another chemical used for blood evidence is luminol. Like other blood test chemicals, it reacts with iron in the blood. Luminol can relieve blood evidence even if someone has tried to clean the blood up. The forensic scientist traces these chemicals on the suspect area and observes the area to see if fluorescence occurs. Luminol has limited use in forensic science in that other chemicals like bleach can produce the same fluorescence as blood. Other agents, forensic scientists use common chemicals like hydrogen peroxide and alcohol in combination with other chemicals at their disposal to produce better test results or speed reaction times. They also can use these chemicals as disinfectants, for instance. Bleach can sterilize work areas or forensic tools. Forensic scientists also may use acid to perform work like revealing etchings on metal. The fact that forensics requires the use of so many chemicals is why anyone interested in a career in forensic science must take chemistry courses. That's all about my topic. Thank you.